Hi everyone. In this last video for the carbohydrates section, we are going to talk briefly about disaccharides, polysaccharides, and the building blocks for RNA and DNA. So let's start with disaccharides. So far we've been talking about monosaccharides, and in particular, we've talked about glucose a lot, so we'll just draw that. But what you can do is connect another monosaccharide to glucose. So let's also draw in our OH groups, and then we'll talk about where we combined these two monosaccharides. So notice that we combined these two through carbon 1 on the first monosaccharide. So that's also the anomeric carbon. And then we used the OH group from carbon 4 on the other monosaccharide to create what's called a glycosidic linkage. Now, because we're using carbons 1 and 4, we can also call this a 1,4 link. Okay. Now, you can link two monosaccharides through other carbons, but this is the most common type of linkage that occurs. You can also convert this particular anomer, where the OH is pointing up, to the other anomer, where the OH group is pointing down. And again, that would be called Muta Rotation. So this is capable of Muta Rotation. So remember earlier we talked about some definitions. So this can basically flip, I'll put it in quotations though, to the other anomer. And that would involve opening up the cyclic structure to the open chain structure and then closing it again. But we can still do that. Now, this particular disaccharide is called maltose. There are other disaccharides. So for instance, if you use different monosaccharides and link them together, you can create other disaccharides such as lactose and sucrose, which I'm sure you've heard of both of those before. Your textbook goes over those in a little more detail, so if you want to look at those, you can. But let's also talk about polysaccharides. So polysaccharides, you can probably guess, is a big long chain of monosaccharides. So these are connected through the carbon 1 and carbon 4 as well. But instead of an alpha bond at the anomeric carbon, we have a beta configuration there. So it's pointing up instead of down, and that will be true for all of these chains here. So that just keeps going, and if you connect enough of them, you can create really useful structural chains. For instance, one of those chains is called cellulose, and cellulose is what provides the structure for trees and plants. Okay, so another one is starch, and there's many other examples as well, but starch is in our food. And these are just composed of these long chains of monosaccharides. But in addition, if we look at cellulose, these polymers can kind of wrap around each other and connect through hydrogen bonds. Because remember, there are OH groups on each of these carbons here. I didn't draw them in just for simplicity, but all of these different monosaccharide units have OH groups that can then connect through hydrogen bonding. So that provides extra structural support. Okay, so now let's talk about our last type of compound called an N-glycoside. So if you start with something like glucose, then you can also perform a reaction that we've seen before. It was a couple weeks ago, I believe, but if you react this with acid and a primary amine, for example, you can create something called an N-glycoside. Now, N-glycosides are the starting point for the building blocks of RNA and DNA. So notice that we just switched this OH group here with an amine group. And if we do the same thing to, for instance, a carbohydrate that looks like this, then we can start to form the building blocks of RNA and DNA. So let's just draw this molecule here. We've got a lot of OH groups pointing down, and then we have our CH2OH group pointing up. Okay, so this is called D-ribose, and we can also react this with acid and some sort of nitrogen-containing compound. Let's see, let me just write here NH2R. And that's going to create something called a Ribo Nucleoside. We can also do the same thing with something called deoxyribo, or 2-deoxy-D-ribose, excuse me. 2-deoxy-D-ribose looks a little bit different. We would replace this OH group here with a hydrogen, and then that becomes our 2-deoxy-D-ribose. And if we react that with the acid and that primary amine, or a nitrogen-containing compound, we can then turn that into deoxyribonucleoside. And there are multiple deoxyribonucleosides, and if you've taken biochem, you probably already know what those are. So let's just kind of talk about those building blocks. So the first type of building block is for RNA. So RNA, again, has that D-ribose as its base. And then we have a nitrogen-containing compound up above. And depending on what that nitrogen group is, you'll end up forming the building blocks of RNA. Same for DNA. So let's draw our DNA building block. So again, we have a 5-membered ring with CH2OH off to the side. We've got an OH group pointing down, a hydrogen, and then some sort of nitrogen-containing compound. And depending on what that is, that's going to give us our building blocks of DNA. So these are really important, these N-glycosides. So for DNA, that nitrogen-containing group could either be something called cytosine, which is, let's see, it looks like this. Oh, whoops, I missed a carbon. Let me draw in another carbon here. And it has an amino group on top. So this is cytosine. So If that group is attached to the DNA building block above, then it's called cytosine, excuse me. But there's also other ones called thymine, adenine, and guanine. So you can look up those structures in your textbook. I'm not going to test you on them. I'm just trying to give you kind of an idea of what this looks like. Let's say we do the same to RNA. Now, RNA does have slightly different structures that attach. So you can use cytosine, adenine, and guanine, but instead of that thymine I mentioned, we would have something called uracil. So RNA building blocks are a little bit different, but you kind of get the idea. All right, so again, I'm not going to test you too much on disaccharides, polysaccharides, or the building blocks of RNA and DNA, but I just kind of wanted to show you the chemistry behind the formation of these really important molecules that really make up all of who we are. We wouldn't be here without them. So that's kind of it for the carbohydrates chapter. In the next chapter, our last chapter for organic chemistry, we are going to cover amino acids, peptides, and proteins. So I will see you then.